Bismillahirrahmanirrahim. Assalamu alaikum. Khanam Durrani here from Department of Microbiology and Molecular Genetics, the Women University Multan. On the behalf of Group 4, today I'm gonna explain the topic Basic Introduction to COVID-19. As we all know that nowadays we all are suffering from a global pandemic named COVID-19. And here as a microbiologist, it's our responsibility to provide a basic knowledge regarding COVID-19. In this video, I'll explain about the structure of coronavirus, history, origin, transmission, pathogenesis, signs and symptoms, prevention, death and cases of COVID-19, and advanced researches of COVID-19. So first of all, let me explain you that what is COVID-19. COVID-19 or coronavirus disease is an infectious disease caused by newly discovered coronavirus, in which CO stands for corona, VI for virus, and D for disease. Formerly, the disease was referred to as 2019 novel coronavirus or 2019 nCoV. The COVID-19 virus is a new virus linked to the same family of viruses as severe acute respiratory syndrome SARS and some type of common cold. It means that the virus is not new, but it is the new strain from family of viruses such as SARS and MRSA. Now let's look at the structure of coronavirus. So coronaviruses are a group of related RNA viruses that cause disease in mammals and birds. In humans and birds, they cause respiratory tract infections that can range from mild to lethal. Here you can see that this is RNA. This is hemagglutinin esterase. Hemagglutinin esterase, HE, it serves as a receptor destroying enzyme, while this is a membrane protein. And these are enveloped small membrane proteins. And these are the spike glycoproteins. Like other coronaviruses, the SARS-CoV-2 genome encodes spikes glycoprotein which protrudes from the surface of mature virions. You can see it in this figure. They play important role in viruses attachment plus in entry into the host cell. Types of coronavirus COVID-19. It has two types. First of all, SARS, which means severe acute respiratory syndrome. And next one is MRSA, Middle East respiratory syndrome. History and origin of COVID-19. So now the question arises that where did the first case of coronavirus disease originate? Retrospective investigations. Retrospective means that looking back on or dealing with past events or situations. So retrospective investigations by Chinese authorities have identified human cases with one set of symptoms in early December 2019, while some of the earliest known cases had a link to the wholesale food market in Wuhan, and some did not. On 31st December 2019, the World Health Organization was formally notified about a cluster of cases of pneumonia in Wuhan city, home to 11 million people and the cultural economical hub of central China. It was all about Wuhan city. The novel human coronavirus disease COVID-19 has become the fifth documented pandemic since the 1918 flu pandemic. The cause of severe acute respiratory syndrome that became known as COVID-19 was a novel coronavirus SARS-CoV-2. The rest is the history, albeit history, that is constantly being written. As of 12 May, 82,591 new cases of COVID-19 worldwide were being confirmed daily and the death rate was over 4,200 per day. Now, let's take a look on coronavirus in man. So, corona is basically a family of viruses affecting respiratory tract, which are causing disease from common cold to pneumonia. And it usually lives in bats and other wild animals, and its transmission to humans directly or via other animals, and it can also transmit between humans via respiratory droplets. COVID-19 infection. So now the basic thing is that what is COVID-19 infection? So, like SARS-CoV, the SARS-CoV-2 virus responsible for the COVID-19 can survive in aerosols for hours and on surfaces including stainless steel, plastic, and cardboard for days. Although washing with the soap or detergents will destroy the virus, it can be transmitted during asymptomatic incubation. Asymptomatic means symptomless incubation phase. This is estimated to occur in 50 to 60% of cases and for up to 2 weeks after the onset of these symptoms. Each person infected passes the viruses to an average of 3 others. The incubation of period is about 5 to 6 days, range 1 to 14 days. Clinical presentation. Clinical presentation varies from asymptomatic subclinical infections and mild illness to severe or fatal illness. Deterioration can occur rapidly, often during the second week of illness. While viral load is up to 60 times greater in people with severe symptoms compared with mild cases. What death? Death is due to pneumonia and possibly hyperinflammation associated with cytokine storm syndrome. Now, what is cytokine storm syndrome? So, cytokine storm syndrome is a type of syndrome in which large number of white blood cells are activated and releases inflammatory cytokines. It has adverse effect on some of the monoclonal antibodies. Hospitalization rates. Hospitalization rates and crude mortality cases in Europe up to 22 April, showing the influence of increasing age, are shown in Figure 1 and Figure 2. You can look in Figure 1 that age-specific hospitalization rates among all reported cases of COVID-19. It is the data from 14 countries in the European surveillance system up to 22 April 2020. And in Figure 2, you can see that age-specific crude case fatality, it means the death per all reported cases of COVID-19. It was the data from 13 countries in the European surveillance up to 22 April 2020. The search starts in Wuhan. The deep root was the search start in Wuhan. An international team of epidemiologists, virologists, and researchers with the expertise in public health and animal health and food safety will lead the World Health Organization's COVID-19 investigation. The agency has not released their names. The team held its first virtual meeting, including researchers in China, on 30th of October, and is reviewing the preliminary evidence and developing study protocols, says the WHO, means World Health Organization. The initial phase of investigations in Wuhan will probably be conducted by the researchers already in China, and international researchers will travel to the country after viewing those results. The agency says, in Wuhan, researchers will take a closer look to the human meat and animal market, which many of the earliest people diagnosed with COVID-19 had visited. With part the market plate the virus's spread remains a mystery. Early investigation sampled frozen animals, carcasses at the market, but none found evidence of SARS-CoV-2. According to a 5 November report of the World Health Organization's mission's term of references, however, environmental samples taken mostly from drains and sewage did test positive for the virus. So preliminary studies have not generated credible leads to narrow the area of research. The report states that the World Health Organization mission will investigate the wild and farmed animals sold at the market, including foxes, raccoons, and sika deer. They will also investigate other markets in Wuhan and trace the animal's journey through China and across borders. The investigation will prioritize animals that are known to susceptible to the virus, such as cats and mink, etc. The team will also look at Wuhan's hospital record to find out whether the virus was spreading before December 2019. The researchers will interview the first people identified to have had COVID-19 to find out where they might have been exposed, and will test blood samples from medical staff, laboratory technicians, and farm workers collected in the weeks and months before December. Looking for antibodies against SARS-CoV-2, the report acknowledges that some of his work might already be underway in China. Plans to look beyond China are sensible, given the extensive surveillance in bats in China. Since the 2002 SARS outbreak has identified only a distant relative to SARS-CoV-2, says Wang. A growing number of experts think that the immediate or close ancestor of SARS-CoV-2 are more likely to exist in bat outside China, says Wang. He says that World Health Organization team should survey bats or other wildlife across Southeast Asia for SARS-CoV-2 antibodies. So the investigation should also prioritize carnivores mammals, farmers for fur, such as raccoon dogs and civets, which had a role in the SARS outbreak, says Martin Beer, a virologist at the Federal Research Institute from Animal Health in Reims, Germany. Beer says that it is surprising that there is no mention of these animals in the report, and we have no information from China about whether these animals have been tested. A spokesperson for the World Health Organization says that the mission will be guided by science and will be open-minded, lucrative, not excluding any hypothesis that could contribute to generating evidence and narrowing the focus of research. So these all are the links of journal and research article from the previous data was taken. Now the next topic is transmission of COVID-19. So COVID-19 direct contact and droplet transmission. COVID-19 is caused by SARS-CoV-2 virus, which spreads between people, mainly when an infected person is in close contact with another. This virus can spread from an infected person's mouth or nose in small liquid particles when they cough, when they sneeze, when they speak, or when they breathe heavily. Direct contact and droplet transmission. So the liquid particles are different sizes, ranging from large respiratory droplets to small aerosols. And respiratory droplets are less than 5 to 10 micrometer in diameter, and droplets nuclei are less than 5 micrometer in diameter. While COVID-19 virus is transmitted through respiratory droplets and contact routes. In an analysis of 75,465 COVID-19 cases in China, airborne transmission was not reported. Oh. Airborne transmission is different from droplet transmission as it refers to the presence of microbe within droplet nuclei, which are generally considered to be particles less than 5 micrometer in diameter and can remain in the air for a long period of times and be transmitted to others over distance of greater than 1 meter. Next is airborne transmission. In the context of COVID-19, airborne transmission may be possible in specific circumstances and settings in which procedures or sport treatments that generate aerosols are performed. Is that endotracheal intubation, bronchoscopy, open suctioning, administration of nebulized treatment, manual ventilation before intubation, turning the patient to the prone position, disconnecting the patient from ventilator, non-invasive positive pressure ventilation, tracheostomy, and cardiopulmonary resuscitation. Aerosol transmission. Next, aerosol transmission can occur in specific settings, particularly in indoor, crowded, and inadequately ventilated spaces, where infected persons spend long period of time with others, such as restaurants, choir practices, fitness classes, nightclubs, office, and a place of worships. More studies are underway to better understand the condition in which aerosol transmission is occurring outside of medical facilities, where specific medical procedures called aerosol generating procedures are conducted. Next is the difference between droplet and airborne transmission. You can see in this figure that droplet transmission, cough and sneeze can spread droplet of saliva and mucus, while in airborne transmission, tiny particles possibly produced by taking are suspended in the air for longer and travel further, less than 5 micron, or these are more than 5 microns, while human hair is 60 to 120 microns wide. Indirect transmission. So indirect transmission through fomites that have been contaminated by respiratory secretion is considered possible. It means that the virus can also spread after infected people sneeze, cough on, or touch surfaces or objects such as tables, doorknobs, handrails. Other people may be infected by touching these contaminated surfaces, then touching their eyes, nose, or mouths without having cleaned their hand first. So COVID-19 mainly spreads from person to person, but it can also be left on objects and on surfaces like lift button, laptop and mouses, pens, digital services, doorknobs, tissues, So if you touch something contaminated and then touch your face and another face, you might at all fall ill. Other modes of transmission. There is some evidence that COVID-19 infections may lead to intestinal infection and be present in feces. However, to date, only one study has cultured the COVID-19 virus from a single stool specimen. There have been no reports of fecal and oral transmission of the COVID-19 virus to date. Asymptomatic transmission. Asymptomatic transmission means that when the infector has no symptoms through the course of the disease, is difficult to quantify. It's very difficult to quantify. Available data, mainly derived from observation studies, vary in quality and seem to be prone to publication biases. While mathematic modeling studies, not peer-reviewed, have suggested that asymptomatic individuals might have major drivers for the growth of COVID-19 pandemic. While similar viral load in asymptomatic viruses, symptomatic cases have been reported, indicating the potential of virus transmission from asymptomatic patients. Pre-symptomatic transmission. What is pre-symptomatic transmission? So pre-symptomatic transmission is when the infected develops symptoms after transmitting the virus to another person has been reported. It means that exposure of secondary cases occurred 1 to 3 days before the source patient developed symptoms. It has been inferred that through modeling that in the presence of control measures, pre-symptomatic transmission contributed to 48% and 62% of transmission in Singapore and China respectively. Pre-symptomatic transmission was deemed likely based on a shorter serial interval of COVID-19 than the mean incubation period, which was 5 days. These all are the links of journal and research article from the previous data was taken. Now, pathogenesis of COVID-19. So, pathogenesis. SARS-CoV, infection of tracheobronchial tree, then infection of immune cells. So it's all about how pathogens transfer. So SARS-CoV, first of all, there is infection of tracheobronchial tree. Next is infection of immune cells, then interferon and cytokines response, and then there is a severe lung injury. This is all about the pathogenesis of COVID-19. Now, pathophysiology. So it is believed that bad coronavirus had acquired the ability to infect human, extending the host range, maybe pangolin, by having a few mutations in the spike protein. And I already told you that what are spike proteins and what is the function in transmission. So the detailed pathogenesis of COVID-19 explained by the following steps. First of all, the viral entry and spread. Nextly, the pathological findings. Third one is acute respiratory distress syndrome, ARDS. Next one is cytokine storm, which I already explained that what is cytokine storm. And the next thing is immune dysfunction. So first of all, virus entry and spread. SARS-CoV-2 is transmitted via respiratory droplets, contact, and potential in fecal oral. Primary viral replication is in mucosal epithelium of upper respiratory tract, nasal cavity, and pharynx. Further multiplication in the lower respiratory tract and gastrointestinal mucosa, giving rise to a mild viremia. Viremia is the viral infection. When viral infection is in the blood, it's termed viremia. Few infections at this point remain asymptomatic, means symptomless. And some patients have also exhibited non-respiratory symptoms such as acute liver and heart injury, kidney failure, diarrhea, and implying multiple organ involvement. SARS-CoV-2 attaches to the host cell by binding its S protein to the receptor protein and angiotensin, converting enzyme 2, ACE2, and then ACE2 is expressed by epithelial cells of the intestine, kidney, blood vessel, and most abundantly in type 2 alveolar cells of the lungs. So the human enzymes transmembrane protease serine2 TMPRSS2 is also used by the virus for S protein, priming and to aid in membrane fusion. And the virus then enters the host cell via endocytosis, and we all know that what is endocytosis. This is the figure in which you can see all the pathogenesis of COVID-19. Next one is pathological findings. So the first report of pathological findings from a severe COVID-19 showed that pulmonary bilateral diffuse alveolar damage. Next, the right lung showed evident dissemination of pneumocytes and hyaline membrane formation, indicating acute respiratory distress syndrome. And the left lung tissue displayed pulmonary edema. Edema is also infection with hyaline membrane formation, suggestive for early phase acute respiratory distress syndrome (ARDS), and the interstitial mononuclear inflammatory infiltrates dominated by the lymphocytes could be observed in both types of lungs. Next thing is acute respiratory distress syndrome (ARDS). So a life-threatening lung condition that prevents enough oxygen from getting to the lungs and into the circulation, accounting for the mortality from respiratory disorders and acute lung injury. More than 40 candidates genes including ACE2, interleukin 10, tumor necrosis factor TNF, and vascular endothelial growth factor, among others, have been considered to associated with the development outcome of ARDS. An increased level of plasma IL-6 and IL-8 were also demonstrated to relate to the adverse outcomes of ARDS. This is the figure which is showing ARDS, means how the T cells are activated and how this pathogenesis occur. Next is cytokine storm. So it is rapid viral replication and cellular damage, virus-induced ACE2 downregulation and shedding, and antibody-dependent enhancement ADE, and are responsible for aggressive inflammation caused by SARS-CoV-2. The initial onset of rapid viral replication may cause massive epithelial and endothelial cell death and vascular leakage, triggering the production of pro-inflammatory cytokines and chemokines, in which loss of pulmonary ACE2 function has been proposed to be related to acute lung injury and further enhance inflammation and cause vascular permeability. This is the cytokine storm, in which the majority of severe cases and death from COVID-19 result from runaway inflammatory responses within the patient's own immune system, causing a cytokine storm that is very difficult to interpret. First of all, normal immune response is triggered. Next thing is macrophages, dendritic cells release cytokine signals. Next, inflammatory reactions of lymphocyte. Next thing is destroying invading COVID-2 cells. Next is lymphocytes damaged while trying to destroy COVID-2. Then damaged lymphocytes release ever more cytokines. Cytokine storm means that release more cytokines, and immune response cycles is out of control, and then inflammatory reaction continue to increase, and this inflammatory response begins to destroy unhealthy cells, leading to acute lung damage. And you can see at this figure then there is an acute lung damage. Next is immune dysfunction. Peripheral CD4 and CD8 T cells showed reduction and hyperactivation in a severe patient, in which high concentration of pro-inflammatory CD4 T cells and cytotoxic granule CD8 T cells were also determined, suggesting antiviral immune responses and overactivation of T cells. While several studies have reported that lymphopenia is basically a common feature of COVID-19, suggestive of a critical factor accounting for severity and mortality. Stages of infection. Stage number 1, asymptomatic stage, initial 1 to 2 days of infection. The inhaled virus SARS-CoV-2 likely binds to epithelial cell in the nasal cavity and start replicating. Next, in vitro data with SARS-CoV indicate that the ciliated cells are primary cells infected in the conducting airways. Next, there is local propagation of the virus but a limited innate immune response. At this stage, the virus can be detected by nasal swabs. Although the viral burden may be low, these individuals are infectious, and the RT-PCR value for the viral RNA might be useful to predict the viral load and the subsequent infectivity and clinical course. Stage 2 is upper airway and conducting airway response. So the virus propagates and migrates down the respiratory tract along the conducting airways, and a more robust innate immune response is triggered. And nasal swabs of sputum should yield the virus SARS-CoV-2 as well as early markers of the innate immune responses. At this time, the disease of COVID-19 is clinically manifest, and the level of CXCL10 or some other innate response cytokines may be predicted of the subsequent clinical source. For about 80% of the infected patients, the disease will be mild and mostly restricted to the upper and conducting airways. Stage 3 is hypoxia. Hypoxia, ground glass infiltrates and progression to ARDS. Unfortunately, about 20% of the infected patients will progress to stage 3 disease and will develop pulmonary infiltrates, and some of these develop will be severe disease, very severe disease. Then the virus reaches the gas exchange unit of the lungs and infects alveolar type 2 cells. Then SARS-CoV-2 propagate within type 2 cells, and the large number of viral particles are released, and the cells undergo apoptosis and die. Suspects areas of the lung will likely lose most of their type 2 cells, and secondary pathways for epithelial regeneration will be triggered. The pathological result of SARS and COVID-19 is diffuse alveolar damage within fibrin-rich hyaline membranes and a few multinucleated guide cell, and the aberrant wound healing may lead to more severe scarring and fibrosis than other forms of ARDS. Recovery will require a vigorous innate and acquired immune response and epithelial regeneration, while elderly individuals are particularly at risk because of their diminished immune response and reduced ability to repair the damaged epithelium. And the elderly also have reduced mucociliary clearance, and this may allow the virus to spread the gas exchange unit of the lung more readily. These are the links of journals and research articles from the previous data was taken. Now, the next thing is signs, symptoms, and prevention of COVID-19. So, coronavirus symptoms are shortness of breath, high fever, vomiting in some cases, pneumonia, cough, diarrhea in some cases. Majority of cases have milder disease, with some having severe disease. Severe cases develop pneumonia and respiratory failure, while deaths have generally occurred in people who are older and who have underlying health conditions. Reports of severe disease in children are uncommon. Do you have a fever? Yes or no? Are you experiencing shortness of breath? Yes or no? Do you have itchy eyes? Yes or no? Are you experiencing shortness of breath? Yes. So you may have COVID-19. Other symptoms may be cough, fatigue, weakness, exhaustion. You may have a flu. Other symptoms have cough, fatigue, weakness, and exhaustion. Do you have itchy eyes? It does not always mean that you have COVID. You may have allergies like sneezing and runny nose. You may have common cold like sneezing, runny nose, mild chest discomfort, etc. Prevention. Prevention is, first of all, wash hands with water and soap or sanitizer at least 20 seconds and avoid contact with sick people. Next thing is, don't touch eyes, nose or mouth with unwashed hands. Don't eat raw food, thoroughly cook meat and eggs. Avoid contact with animals and animal products. If you are infected, what to do if you are infected? First of all, stay at home. Because stay home and stay safe. Avoid contact with others. Cover your nose and mouth when sneezing. Keep objects and surface clean and wear a surgical mask, which is N95 respirator. COVID-19 prevention. The first and foremost prevention of COVID-19 is safe distancing. Maintain a social distance of about 6 feet. COVID-19 prevention. Travel advice. Avoid traveling to affected areas unless necessary. Make sure you all have necessary vaccination and travel medication. Seek advice from your healthcare provider. Don't travel if you have fever and cough. And if you have become sick while traveling, seek medical care immediately. Coronavirus prevention. Stay at home and try to do more work from home and avoid contact from infected people. These all are the links of journals and research articles from the previous data was taken. The next thing is COVID-19 death and cases. So, coronavirus first case origin. December 31, which I already told you in history and origin section that in December 31, China alerts World Health Organization to several pneumonia cases. At January 7, France confirmed Europe's first coronavirus case. January 11, China announces first death from coronavirus. January 13, 2020, World Health Organization reports cases in Thailand and first outside China. January 30, China alerts World Health Organization to several pneumonial cases. February 22, 2020, first death outside China recorded in Philippines. At February 7, Chinese doctor and whistleblower Li Wenliang dies. At February 14, Egypt confirmed Africa's first case. And at February 11, World Health Organization names viruses COVID-19. You should remember that February 11, World Health Organization named this virus as COVID-19 and it was originated at 2019. The next thing is COVID-19 disease or death and cases. So in USA, you can say that USA, India, Iran, Pakistan, and 4 Iceland and Tonga are affected, in which these are the case cumulative total, and these are case newly reported in the last 24 hours, and these are death cumulative total, in which you can say that USA is highly affected, and these are the deaths newly reported in the last 24 hours. While if we talk about transmission classification, that in USA there is community transmission. India and Iran, in India there is cluster of cases of community transmission, while in Pakistan there are cluster of cases, while in 4 Iceland there are sporadic cases, while in Tonga there are no cases. These are the cases rate worldwide and death rate worldwide. Evolution of the amount of cases. Evolution of confirmed COVID-19 cases in the world: November 2839, December 2475, while January 2123. Countries with the highest infection rate. So there are the countries with the highest amount of confirmed cases over time includes United States, Spain, Italy, and China, where this virus was firstly originated. Now, total recovered people from COVID-19 as of January 2021 was 50,958,070. These are the references or links and journals from the previous data was taken. Now the next thing is advanced researches and recent evidence regarding COVID-19. So first of all, the evidence published between 4/11/2020 means 4 November 2020 and 10th November 2020, that a prophylactic nasal spray. A team from Columbia University has released preliminary result, not yet peer reviewed, showing that the intranasal administration of a lipopeptide that binds to the virus spike protein completely prevents SARS-CoV-2 infection in ferrets. These lipopeptides are inexpensive, highly stable, and non-toxic. If they work in humans, they could be used as prophylactic to prevent infection by SARS-CoV-2. A drug from rheumatoid arthritis. Artificial intelligence analysis have identified baricitinib, an immunosuppressor used for the rheumatoid arthritis, as a potential drug for COVID-19. And in a small trial in hospitalized COVID-19 patients, the drug reduced inflammation, progression to severe disease, and mortality. While antibodies in breast milk from COVID-19 recovered mothers contain antibodies that can neutralize the virus, especially IgA antibodies, and that could protect the babies from infection, according to a study. Understanding the loss of smell. So what is the concept behind understanding the loss of smell? So cells that provide support to our olfactory neurons express receptors that allow SARS-CoV-2, providing the virus with a potential entry door into the brain, according to a study that analyzed gene expression in the olfactory system. While accordingly other study, not yet peer-reviewed, reveals viral presence in olfactory mucosa of COVID-19 patients who lost their sense of smell. Convalescent plasma. Next thing is convalescent plasma. So a controlled clinical trial of convalescent plasma for the treatment of COVID-19 severe pneumonia showed no benefit in terms of clinical outcomes or mortality, despite the fact that the levels of IgG antibodies to SARS-CoV-2 were controlled before transfusion. Evidence of early circulation. An Italian team found SARS-CoV-2 RNA in a throat swab collected from a child in early December 2019, around 3 months before the first identified coronavirus disease case in Italy. So the child had not traveled. So this finding raises the possibility that the virus was circulating outside China earlier than thought. Impact on male fertility. So is COVID-19 have impact on male fertility? So a small study on testis biopsies from COVID-19 patient revealed that virus can infect germ cells and affect the production of spermatozoids. So these findings raise the possibility that the virus may affect the male fertility. No benefit for an antibiotic. The UK recovery trial found no benefit from azithromycin in patients hospitalized with COVID-19. Pfizer and BioNTech. The final phase 3 result from the Pfizer-BioNTech vaccine were published this week. The two doses given 21 days apart were safe and 95% effective against COVID 19. Mixing vaccine. A trial may start soon to find out whether mixing COVID 19 vaccines give better protection than two doses of the same one. So, last but not the least, I would like to conclude my topic about basic introduction to COVID 19. First of all, avoid social gatherings, theaters, etc. Next thing is cancel non essential travel for few days. Kindly explore work from home opportunity. Consider distance learning or e learning. Short term holidays of school, no problem. Stay home when sick and limit visitors. Limit large work gatherings like meetings and trainings and consider structuring work schedules at offices and stay abreast with development related to the epidemics. These all are the references or links of journal and research article from the previous data was taken. Thank you so much for your kind attention. I hope that there is no query regarding introduction of COVID-19.